The macula of the utricle and the saccule are covered by a small patch of hair cells. Each cell is positioned in a slightly different orientation. This distribution basically covers all orientations in 360 degrees. The tip of all the hair cells are covered and in contact with a gelatinous mass which has also embedded within it dense particles made out of protein and calcium carbonate known as othalites or statoconia. The combination of a gelatinous mass with heavy particles incorporated permits that this mass, following the law of inertia, tend to stay in place when the head starts to move and tend to keep traveling when the movement of the head ceases. As a consequence of this phenomenon, when an animal starts to move, the stereocilia and kinocilium initially bends in the direction opposite to the initial movement. Then it catches up with the movement and straightens up, and when the movement stops, this must bend the stereocilia and kinocilium in the direction of the movement. Since all stereocilia and kinocilium in the macula bend in the same direction, but each cell is placed in a slightly different orientation, then a given movement may result in a full depolarization of some cells, in partial depolarization in other cells, and or in full hyperpolarization on others. The opposite occurs when the movement stops. Given that every cell connects to the central nervous system, the central nervous system recognizes the position of each cell, and by analyzing the degree and speed of depolarization or hyperpolarization resulting from a given movement, the central nervous system can identify the precise direction and speed of that movement. Full hyperpolarization